This is going to be for Chapter 3, Part 2. I'm going to pick up where we left off with the cell membrane and then move into passive and active transport. All cells have a cell membrane. They control what enters and leaves the cell to help the cell maintain homeostasis. That's to maintain a stable internal condition. Think of it as the skin of the cell. It's going to provide protection and support for the cell, just as your skin is your body's first line of defense for protection and support of your body. Okay, cell membranes are composed of two layers of phospholipids. That's called the phospholipid bilayer, where bi means two. So 2 layers of phospholipids. Each layer has a phosphate head and fatty acid tails. In the middle, it's glued together by a glycerol. And so if you see this picture right here, the yellow circles are the phosphate heads. They're pointed on the outside, with the fatty acid tails pointed on the inside. Just like you see in these pictures. The polar heads. The phosphate heads are polar, meaning they have a positive charge and a negative charge. The fatty acid tails are nonpolar. They repel water. Okay? Think of it as a peanut butter and jelly sandwich. The bread is on the outside, the peanut butter and the jelly is on the inside. In this case, the phosphates are on the outside, the fatty acids are on the inside. Now, the phospholipid bilayer, the cell membrane, also has proteins embedded, and these proteins do a lot of different functions, from being transport proteins, or like right here, you see that they have carbohydrate markers attached to them. That means that the cells are able to recognize other cells or do specific functions, like recognizing certain chemicals or materials are floating around the cell. Now, these cells have pores. Because the phosphate heads are kind of circular, that means there's space that materials could just go straight through the cell membrane. Now, the cell membrane is selectively permeable, meaning it has selective permeability. It allows certain materials to enter, but it keeps other materials out. You can think of the cell membrane as being like the bouncer to a club. The job of the bouncer is to kick certain people out, but also be really picky or choosy about who they let into the club. And that's exactly what the cell membrane's doing. It's going to be picky. It's going to be choosy about what it lets into the cell and what it lets out of the cell. So again, here's a close-up picture of it. So here's a phospholipid, circular yellow phosphate head with the fatty acid tails, 2 layers, lipid bilayer, 2 layers of the phospholipids with their fatty acid tails pointed in. We've got transport proteins. We've got proteins with carbohydrate markers. Now we're going to talk about passive and active transport. Diffusion is a type of passive transport. It doesn't require energy. Moves down a concentration gradient until it eventually reaches equilibrium. Equilibrium is where there's an equal distribution of solutes, so an equal distribution of materials. We need to compare and contrast passive versus active transport. Passive transport does not use energy, so no energy. The 3 types are diffusion, which is also known as simple diffusion, facilitated diffusion, and osmosis. Active transport does require energy. Examples are protein pumps, endocytosis, and exocytosis. Passive transport goes from a high concentration of something down the gradient to a low concentration of something. Active transport is just the opposite. It's starting with a low concentration and is working up to a high concentration of something. Good examples, like what you see in the picture here. Pretend you are on the top of a very, very, very steep hill. You are in roller skates. It does not require any energy from you to go from being very high up that hill to skating down to the bottom of that hill. You just kind of roll. But with active transport, because you've actively got to work your way up that hill, that's going to require energy. So if you are at the bottom of a very steep hill in roller skates and you have to skate up that hill, it's going to take a lot of energy. I mean, think about it. If you've ever skated up a hill, biked up a hill, or even walked up a hill, by the time you get to the top, you are winded You might be sweating and you have just used a lot of energy. Well, that's what's going on with active. Passive, again, you're just gliding down the hill, high to low, no energy. Active, you're at the bottom of that hill having to work your way up from low to high. Because you're going from low to high, you're going to be using a lot of energy. In passive, no energy, molecules are going to move randomly in this case, from a high concentration to a low, where there are three types: diffusion, facilitated diffusion, and osmosis. Diffusion is going straight through the membrane. That's why it's called simple diffusion. It's going to start with a high concentration. So then we're going to pretend that like this is a glass of water and this is a sugar cube. You drop a cube of sugar in water, there is originally a large concentration of sugar molecules right here. And eventually it's going to diffuse or dissolve out. And once it reaches the level where it's fully dissolved or diffused, it's going to be evenly spaced throughout the solution. That's how diffusion works. Facilitated diffusion. To facilitate means to help. So facilitated diffusion is help diffusion, and it's helped through transport proteins. If you look at the picture, B right here represents regular, standard, simple diffusion. These little red molecules would go straight through the pores in the cell membrane. But with facilitated diffusion, these molecules might be too large or they might be charged, but they need a little extra help to get across the membrane. And that's where the transport proteins come into play. This specific type of transport protein is called a channel protein. It acts like a bridge for it. So, around here, our bridges, they don't normally move, okay? Because they don't normally move, you just drive straight over them. Okay, that's what's going on with the channel protein. These proteins do not move. The molecules go straight through them. Now, this picture right here called a carrier protein is another type of transport protein, but that is in protein pumps, active transport, because this protein is going to have to physically change shape to pump materials in and out. But little channel proteins are type of transport proteins that don't require energy and are going to be involved in facilitated diffusion, type of passive transport. Here is another example, just a closer up of it. So facilitated diffusion. This is outside the cell. This is inside the cell. Outside the cell, there's going to be a high amount of glucose or sugar molecules compared to inside the cell where there's a low amount. Because these molecules are going to move from high to low, this molecule is going to go straight through the channel protein into the cell. Again, this channel protein is a type of transport protein that does not change shape. Therefore, that is passive transport and facilitated diffusion. Osmosis has to deal with the diffusion of water. Water is such a special molecule that it has its own name for the diffusion of it, osmosis. Water moves from where there's a lot of it to an area where there's not a lot of it, and it's going to go straight through the cell membrane. We're going to talk about the three osmotic solutions in just a few minutes.
To the office, Miss Mullins.
Sorry for that. With active transport, the cell uses energy.
Cancel that, Miss Mullins.
With active transport, the cell uses energy. Molecules are actively moving to where they need to be. The cell is typically changing shape, and because it changes shape and structure, it requires energy. We're moving from a low concentration to a high concentration. The first type of active transport are protein pumps. These proteins are going to physically change shape. They're going to move. I wish this animation showed. They're going to move. So an example is the sodium-potassium pump. 2 sodium ions are going to attach here. 1 potassium ion is going to attach here. Once they are in place like a lock and a key, this protein is going to twist around, and then the potassium is going to pop out, and the sodium is going to go in. Endocytosis. Endo means into. The cell is bringing something into it. Okay, this because the cell is physically changing shape, it's requiring energy. Each of these three examples right here are known as cell eating or phagocytosis. This is an amoeba. This amoeba is trying to eat this paramecium. Is extending out around it to where it's going to fuse together to form a vesicle that will eventually be digested away by a lysosome. So here's another picture of it. It's going out and around, stretching out and around it, and it'll form a vacuole. This is how our immune system gets rid of certain things like bacterial infection. So this is a white blood cell reaching out and around a bacterium. Now, this is not how our body battles viruses. That's different. Okay, but this is how our body digests away a bacterium. Exocytosis. Exocytosis is going to force material out of the cell. Okay, so this is how our cells secrete hormones or how they just get rid of wastes. So this vacuole that's going to be filled with a hormone or waste is going to eventually work its way to the cell membrane, fuse with it, and then release its contents. It's gross, but the best way to think about this is a pimple. Pimples are form under the skin, and then you pop them, and all the stuff comes out. That's exocytosis. Endo, into. Exo, exit.